In this 2-minute webinar, we'll see how to find information about a particular gene. The first thing we want to do is select as our currently searchable database the organism in which we want to search for a gene. Right here it tells us that our currently searched database is the rat. Let's change that database to Bacillus subtilis 168, reference genome. So we now get a summary page describing this organism. Let's now search for our gene of interest, which is DEOD. And when we do this search, we only get one result, which is the gene page for this gene in B. subtilis. And so it shows us the name of the gene and the gene product, accession numbers, the length of the gene and its product, reactions the gene catalyzes, and the pathways in which those reactions are present. We also see links to other databases. And these different tabs up here let us see other information. For example, each individual reaction is broken out in this screen along with the pathway or pathways it's present in. And this screen shows us protein features on the amino acid sequence. So now we can also perform a more elaborate search using the tools search menu to search for genes, proteins, and RNAs. For example, we can search by the sequence length. We can search for all proteins in B. subtilis that are 50 amino acids or fewer. And there are many other options you can explore here, from searching by the PI of the gene product, the product type and subunits, etc. Let's submit this query, and we get a table of results initially sorted by gene name. We can sort it by length by clicking right here. We get an ascending sort on length. Let's do another search. Let's search for sporulation. And we get a number of different results. We can see there are protein results, RNA results, and gene ontology results. So these are all the proteins that have sporulation in their name, and quite a few. There are also RNAs that have sporulation in their name. We can click, for example, on this one, and we'll go to the gene protein page and see, for example, references for this gene. And if we click on the Essentiality tab, we'll see that when grown in LB medium, if this gene is knocked out, the organism does still grow.